Kristen Atchison here, and we are still talking about Chapter 3, the visual brain. This is our second lecture about the primary visual cortex, which we also call V1. So on this image of the brain, V1 is that tiny little yellow area in the occipital cortex. Remember, your occipital cortex is the one in the back of your head. If you rest your hands on the back of your head, you're resting your hands on top of your occipital cortex. And again, that little yellow part down at the base, where the two hemispheres meet, is the primary visual cortex. This is the first stop for visual information, is the primary visual cortex, which is how it gets its name, okay? That's because it's the first stop. After that, the information can go into different areas depending on what kind of information is, including the occipital lobe, the temporal lobe, and the parietal lobe. And the farther along the information gets, the more complex the information gets, the more specific it gets. But the primary visual cortex is the first step in the brain from the photoreceptors in the eye. So again, this area V1 is really pretty small. It's only about 2 millimeters thick, and it covers only a few square inches of the brain. And like I said, it gets its name by being the first area to receive that visual information. Now this area, despite how tiny it is very, very highly organized. And it's organized in a number of different ways that we're going to talk about. The first of which is that it's organized into cortical columns. So basically, a column is something that holds up a building, right? So it's that vertical kind of core sample. So you can think of it as kind of a core sample of the brain, that it's these cortical columns. It's also organized renotopically, which we'll talk about in other slides, and we've talked about a little bit already. And that's the idea is the organization that's in the retina transfers over to the organization that's in V1. So if receptive fields are next to each other in the retina, those receptive fields are going to, the area of the brain that processes those receptive fields are going to be next to each other in the cortex. And the other thing that it shows is cortical magnification, which again we'll talk about more, but it's basically what happens with convergence. So convergence is when, again, we have multiple photoreceptors going into one RGC receptor or one RGC cell, or just one photoreceptor going into one RGC cell. And that's going to transfer over to how much space in the brain it's taking up. If you have a whole bunch of cells going into one RGC, it's not going to take up much area in the brain. Whereas if you have one cell going into one RGC, it's going to take up a lot more area in the brain, and we'll talk about that more. There's different kinds of cells found in V1, simple cells, complex cells, and end-stop cells, and each cell responds to progressively more complicated stimuli. So our simple cells are going to respond to the most basic, simple kind of information, hence its name, right? The complex cells are going to be more complicated, and so on and so on. So a lot of this research comes out of the researchers Hubel and Wiesel, and they really did the first systematic studies of cortical neurons. And like most scientific discoveries, they found some of this by accident. They thought that they were looking for one thing, and just by accident, they found that these cells were actually responding better to edges. And so they found that they are most effectively stimulated by bars or edges with really narrow ranges of orientation. And when we say orientation, we're talking about the angle at which this bar is placed at. So is it a straight line that goes just from left to right, or is it an angle? Is it a vertical line? Kind of depending on the cell, depends on which angle or which orientation that it really prefers. And this they found for both simple cells and complex cells. And they actually won a Nobel Prize for this work in 1981. There's some videos that are posted on Canvas that you can watch, as well as the YouTube playlist that show you kind of this research. They did it with cats and some monkeys. But it's really, really interesting stuff and famous stuff. So if you don't already know about it, I encourage you to, because I think it'll help you understand what we know about the cells in V1. So as I said, the cells in V1 are organized renotopically. They have renotopic mapping. And that's the idea that the map in the retina matches the map in the cortex. Okay? So if you have receptive fields that are next to each other in the retina, we're going to have the area of the brain that processes those receptive fields next to each other in the cortex. And we see this in terms of their columns. So if two columns are next to each other, they're going to have receptive fields that are next to each other. And how we map this renotopic map, right? When we talk about maps, we talk about north, south, east, and west. We have coordinates for renotopic maps as well. We call these polar coordinates, and there's two of them. The first is eccentricity, and that's the distance from the center of the fovea. Remember, your fovea is when you're focusing on something, you're putting it on your fovea. So eccentricity is the distance from how far into the periphery is it, essentially, right? Because you're fixating on something on your fovea. So how far into the periphery is this? Is it really close to the fovea, or is it way off in the edge of your vision? Polar angle is the angle above or below the horizontal in a circle with the fovea at the center. So this is going to be more, a little bit more 3D. And so it's going to not only be kind of the distance, but it's going to be the angle, okay? So eccentricity is going to be kind of how far it is, where polar angle is going to be more about the angle it's at. And these are measured in different ways. So in an fMRI study, this is the image from your book. It's image 315 on page 101. You can see that they have the eccentricity map, and the visual stimuli that they used to measure that was kind of this checkerboard pattern around the fovea. So they would be focusing on the middle of that X in the middle, and then they would measure activation kind of around it. While as for the polar angle, again, they're going to be fixating at the very center there, kind of at the peak of that checkerboard. But as that moved around, they could measure activation as well. And what they found, again, is when these things, the receptive fields are next to each other. So if it's to the left of the fovea, it's in your left visual field, it's going to be activation is going to be to the left of that in the brain. So again, that these things are the maps match up, right? So a map of campus is a representation of actual campus, right? The idea is that, and why it gets to the name map, is the idea is that the map that you get in the cortex of where things are is a representation of them in the visual field. And that because it works that way, it's because of the representation in the retina. So here's an example of that. So when they look at one cortical column, and they look at that the activation of one cortical column, which is what we're seeing in image A, we see that because they are on top of each other, the receptive fields are on top of each other. These receptive fields all overlap. So when they stick in an electrode, and they just stick it down one cortical column, all those receptive fields are going to overlap in the visual field. So here's a picture, and we have George Washington there, and all those receptive fields are kind of on George Washington's face, okay? Whereas if they stick the electrode in at an angle, so it goes through several cortical columns, which is what we're seeing in image B, the receptive fields may overlap a little bit, but they're all adjacent. They're all next to each other. So you see that the red neuron and the red receptive field match up, and the green neuron and the green receptive field match up, and the purple neuron and the purple receptive field match up, and the blue and the blue. And you see that you have that same progression. So you see that green is to the right of red, and purple is to the right of green, and blue is to the right of purple. So again, we really have this map in the cortex of the retina and subsequently the visual field. Another thing that's going on, and another way that the primary visual cortex is organized, is called cortical magnification. And so really what this means is that there's not a one-to-one relationship in terms of the cortex. We don't see that the area on the retina is equally represented in the cortex. So while we have a map and we'll see these things adjacent, it's not going to be an equal representation. And that's because the amount of cortical space is related to how many RGC cells are covering that space. And remember convergence. Remember how we have multiple photoreceptors in convergence when something's highly convergent going to one RGC cell. And when we have no convergence, we have one photoreceptor going to one RGC cell. What that's going to translate to is because those RGCs have axons, right? One axon will carry a smaller receptive field if it only has one photoreceptor, where it's going to carry a much larger receptive field if it has multiple photoreceptors. What that translates to is in the brain, how much space it takes up, right? How much information is going there. And so we have an image that's from your book. This is image 316, again on page 101. The image on the left is the left-right visual field. And this is kind of how you would see it. So if you were looking at a pattern of these different colored dots, and you were looking at the orange dot, that's what you're looking at, or right next to the orange dot, that center of the fixation. We're only being shown the left visual field here. And so then in the brain, we're only being shown the right hemisphere's V1, okay, because again of our contralateral organization. So we have the left visual field, and you see in the visual field, those dots are all the exact same size, right? Our fixation point is our fovea. Remember, our fovea is really highly densely packed with photoreceptors. It's got cones upon cones upon cones upon cones, right? Whereas the further we get into our periphery, the less cones we have, the more rods we have, and the higher convergence we have is to the further we get into our periphery. At our fovea, there's very little to no convergence at all. And so when you look at the cortical map of the brain, you look at renotopic mapling, we're still seeing those colors are next to each other, right? We're still seeing that orange is next to yellow, just like it was in the visual field. We're still seeing that green is next to blue, just like it was in the visual field. So we're getting that renotopic mapping. But what we're also getting is cortical magnification. Because the fovea has more photoreceptors and less convergence, it takes up more space in the brain. So that little orange dot that is the same size as every other dot in the visual field is huge amount of space comparatively in the cortex. What that really does is allows for better acuity. Remember, acuity is how well you can see. And so when you're focusing on something, you really need high levels of acuity. You need to be able to find detailed information about the thing that you're staring at, right? When you're reading a book, you need to be able to see all the differences in all the letters. You need to be able to see where the spaces are, where one word stops and where another one begins. You need that level of acuity on what you're focusing at. And so, again, cortical magnification really allows for high levels of acuity because of this lack of convergence. So, again, remember the fovea has very highly dense RGCs, and they have really small receptive fields. And because of this, and again, we have that decline as we go out to the periphery, right? And because of this, we have little to no convergence in the fovea itself. And so that really allows a lot more area of the cortex to take up the receptive fields of the fovea, whereas our periphery is going to have a lot less cortical space dedicated to it. It's still important. We're still getting information about it. We're just not getting as much information as the information we're focusing on. And that will really lead us into talking about attention later on in the text, later on in the course. That where our focus is different than something where our focus is not, and how that will play out. And we'll discuss that later. So again, to kind of just sum this up, our RGCs on our fovea have really small receptive fields, and so they have tons of V1 cells to cover that area of the fovea. Whereas our periphery, we have much bigger receptive fields, and so there are much fewer cells needed to cover that area. So this will lead to that trade-off between acuity and sensitivity. So we're going to have better acuity at our fovea, but because we have more cells in our periphery, we're going to have better sensitivity. But we're not going to have as good of acuity. So there's this trade-off between acuity and sensitivity. And this is played out in area V1. So this ends our discussion about V1 for our second lecture for chapter 3. Thanks.